It's my great honor and pleasure to introduce our next speaker, who is connecting to us from the US. It's Dr. A Kesterson. He had a long career in genetic research, including extensive work studying the mechanisms of gene transcription and regulation. Dr. Kesterson is currently leading several NF1 research and gene therapy projects. Bob Kesterson studied at Baylor College of Medicine and was a professor at the University of Alabama in the Department of Genetics for many years prior to his move to LSU. I'm really happy that you are joining us, and Bob Kesterson will give an overview of gene therapy projects in NF. Thank you for being with us. The floor is yours.
Thank you so much, Klaus, for inviting me to give this presentation. What I really want to talk about is what goes on with NF1 at the cellular level. So in order for us to understand gene therapy, here's a cartoon showing a cell, and then there's a nucleus, and then these are, a cartoon of the chromosomes, and chromosome 17 is where the NF1 gene is located. And so every cell in your body inherits, one chromosome from your father and one chromosome from your mother. And so you have two copies of the NF1 gene. And then as was likely presented in early morning presentations, the defects that arise is, you inherit one bad copy from your parent, or 50% of the time, it's a brand new mutation that happens within the embryo. And so then, and this first hit is called the constitutive mutation. And so a cell is then challenged. It only has 50% or half of the normal amount of gene function. And so it can still make neurofibromin, but it doesn't function normally. And then the real trouble occurs when you get a second hit. And then that second mutation then leads to what's called loss of heterozygosity. So now you don't have either gene that works. And so in the end, you don't make any neurofibromin, and that's bad for a cell, and it's true for other tumor suppressors, and that the cells then start growing rapidly. And so this is the cartoon of what's going on. And then really to understand what we're trying to do with gene therapy, we're trying to, based on the kinds of mutations that occur, and to put this, to remind everyone, DNA operates in an interesting way in which the DNA is read in groups of three, and the groups of three then make amino acids that then get strung together to make a protein that's needed. So the type of mutations that can occur can lead to large deletions, so you completely delete the NF1 gene, or you can have very small changes in sequence, and just a change of one nucleotide, one base pair, can lead to the wrong amino acid being made. Or uniquely in NF1's case, about 20% of the time, there are signals that tell you when to start reading a gene and when to stop reading it. And 20% of the time for NF1, you get these premature stop signals. So they tell the gene to stop too early, so you don't make a full-length protein. And so there's all these different kinds of gene therapy to try to edit and change back what we're doing. And I'm going to try to give you examples of each of these, and even talk about CRISPR. That's kind of the holy grail now. It's been in the news for the last 10 years, which can we change so we simply correct the DNA so that it's back to normal again, and therefore the gene will function okay. If you haven't seen this slide before, it is really, I think, a remarkable summary slide, really depicting how complicated the disease associated with NF1 is, how the same issues that we have for NF2, SMARTB1, LZTR1 on the right-hand side. So it is remarkable the number of systems and the different cells within the body that are affected by the loss of these genes. And it really, what I tried to, when I'm explaining this to the people first joining my lab, I said, it's when and where you get that second hit in the NF1 gene that then causes the specific phenotypes or the disease presentation that you see. If you get a second hit in your certain blood cells, then you end up with leukemia. If you get a second hit in mammary tissue, you get breast cancer. And we now know that breast cancer is a major issue for NF1. If you get, it could be that just the first hit is enough to cause problems with cognitive development. We're not sure just yet, but there are a lot of experiments heading that way. For the common kind of plexiform tumors or cutaneous tumors, if you get a second hit in the cells that are responsible for wrapping large nerves, then that can give you these large plexiform tumors that become problematic and can lead to peripheral metastatic tumors. Or if you get a skin-specific hit, then you get cutaneous neurofibromas, and and so on and so on. You can have bone defects. You can have a whole lot of different types of tumors associated with loss of NF1 gene function. And a similar story with the NF2 related disorders. It's when and where you get that second hit. So when I think of gene therapy, what we try to do is, this is kind of this big wheel, this cartoon of how we approach this for considerations in preclinical development. What do we do with our, how do we approach this with our research tools we have at our disposal? And so we have to think about the type of mutation that the patient has. Is it a large change? Is it a small change? And really, what does that affect? How does the mutation affect the function of the gene? And then we can create cell culture models, or we can create animal models. We create mice, we create rats. There's now zebrafish that our group has worked to create. And then we also have pigs that have been modified. And what we're trying to do is introduce the same type of mutation into these animal models or in our cell lines. And then we apply the different or the appropriate type of gene therapy that then could restore function to the gene. And so I'm going to give you some specific examples of that. And then, well, the challenge that we really haven't addressed much yet has to do with, well, once we create a system for correcting the gene, how do we deliver that? And then we have to work on optimizing the route of gene therapy. As I said, if the problem is in the bone, it does us not a lot of good to try to do a skin-specific application of our gene therapy. Now, if we have cutaneous tumors, then maybe our skin applications will work just fine. But we have to work on how do we introduce the drugs that we develop to then target the right tissue. As I mentioned, there's all different types of mutations that can go wrong. And what we're trying to do, and what we have been successful in, is developing animal models that represent every different type of mutation. And I want to try to give you some specific examples of how we then use these models. And these are all, what's shown here are patient-specific mutations that have been recreated in animals, and that we're then deriving specific therapies to try to correct those. This is a cartoon of the NF1 gene. It's a rather large gene. And this is just, the slide's a little bit old, but this is showing where the different mutations that we're trying to reproduce in animals. Here's a list of the different mice and rats we've made. What I failed to tell you is that the NF1 gene is very complicated. It's large, and we already know it has over 7,000 known mutations. I mean, you'll see in the literature 3,000 or 4,000, but Ludwig Messiaen did a remarkable job and has this very large database that she created at the University of Alabama at Birmingham, and characterizing over 7,000 mutations that exist. So and that's really different than other diseases that may have one common mutation. If you think about cystic fibrosis, for example, about 70% of the patients with cystic fibrosis, they have the same mutation. It's a simple delta, a phenylalanine that's been deleted. So that in and of itself makes it much more difficult to study NF1 and to develop therapies. So one of the therapies I'm going to tell you about is nonsense suppression. And the reason for that is, for most genetic disorders, about 10% of the time, you'll have these what's called premature stops, where the mutation has told the gene to stop making, there's always a start and a stop. And the nonsense mutations tell it to stop too early. And 10% of the time, mostly for genetic disorders, there's nonsense mutations. But for NF1, it's actually 20% of the mutations. So if you think about that, represents 1 in 5 NF patients that have these premature stop mutations. And I've been working and made several models to address this, made preclinical models in Hurler syndrome and cystic fibrosis and other diseases with a group at UAB for many years. And so the first animal models we made were premature stop mutations. And the reasons for that is, so this cartoon shows you, if you're seeing my pointer, let's say the start of the gene is up here, and then the normal stop is down here. So then that would make a full-length protein. You know, every 3 nucleotides, you'd insert an amino acid, and then the protein keeps growing and growing. But then if a mutation comes along, then it makes a signal to stop too early. And then, so as the protein is being synthesized, it sees this premature stop, and it just falls apart. So now you end up with a truncated protein, and it doesn't work. But we have drugs that will trick this ribosome, so the part of the machinery that's making the protein, we can trick it into sticking a different amino acid in there. So it's reading this premature stop, but now it sticks a different amino acid in there. Sometimes it sticks in something that the protein won't work, but a lot of the times it can stick in the normal amino acid or another amino acid, and that allows the gene to work normally. And so again, this is tried and proven therapies. And in fact, these are a list of diseases here where there's multiple clinical trials for this nonsense suppression therapy to work. I'll say if you live in Europe, then you're lucky, because Duchenne's muscular dystrophy actually has a drug called atalurin that will work to suppress some of these nonsense mutations. So this is something that I think has a promise for NF1, mainly because these are small molecules and you can deliver them easily. You can give them orally, and they can go to different parts of the body and restore gene function. One of the first mouse models, in fact, that we made was this patient mutation that's shown here at this position 681. And this is just a premature stop. And in the mouse, we have to do some tricks in mice in order to bring it to mimic the second hit. But we can create plexiform tumors along the spinal cord in mice. And you can see this is a neurofibroma that's pinching on the spinal cord. And so this is really an accurate representation or recreation of what happens in patients. And then these are just, figures of what's characterizing the effects on the nerves. So if you think about how do we do then clinical trials, and again, what we're trying to do are mouse clinical trials, because in the end for gene therapy, we have to have convincing data that, one, we can restore gene function, and two, that it's going to be safe. And so we really develop these animal models to then prove to the oversight agencies that are going to allow you to do clinical trials that we have a good therapy that we think will work. And so we can do things like a prevention therapy. If you think about it, we could take animals that are going to be affected and put them on drugs soon after they're weaning and just look, well, can we prevent the tumors from forming? In our model, the tumors normally form around 4 to 5 months of age. So it takes a while for us to develop these animals and for them to show tumors. Or we can do what's called a treatment protocol. That is, wait till tumors arise, image them, and then put them on drugs. And this is really what you would be doing in a true clinical situation anyway. You'd wait until a patient had tumors and enroll them, and then look for, can you interrupt the tumors? And so we have ongoing clinical trials with our mouse models now. We're well funded by the Gilbert Family Foundation. That's a family that's affected by NF1 mutations. And so we are looking at multiple ways in clinical trials with this now. So another approach that you can think about as gene therapy is, so I mentioned that the NF1 gene is large. It actually has 55 exons. So it's a very large gene. It makes a large protein. So there's a lot of opportunity for things to go wrong. In fact, I think that's why it's more prevalent than other diseases. It's simply, it's large, and there's this chance for hits to occur. My colleague at UAB, Deanne Wallace, who's a very close collaborator of mine, she's been pursuing this idea that there's certain regions of the NF1 gene that maybe we could do without. It's such a large protein, it has multiple domains or regions that can function. Well, can we skip over certain parts of it and still make a protein that then works? And so in this cartoon you see here, suppose we had a patient with an exon or had mutation in exon 4. Could we then apply what's called an exon skipping approach using antisense oligonucleotides? Again, ASOs, this is a proven gene therapy approach that has been worked in Duchenne's muscular dystrophy, spinal muscular atrophy, hypercholesterolemia. I mean, there's probably another 20 diseases where ASOs are already in clinical use or have been approved. And so in this case, we could take an ASO and cause the gene to skip over this bad exon. And so it splices, let's say in this cartoon, 3 to 5. Now you get a smaller protein that's made, but it functions. And so then that's one way of thinking about a different approach to restoring gene functions. And now I want to share with you another use of ASOs. About a little over 10 years ago, Klaus, I was hearing your story, and honestly, I've worked very closely with Joseph Kammermeyer in Germany in the Nothing is Forever Foundation, and I'm sure you know Joseph, and so we collaborated. He approached me. He was one of the first foundations to approach us when he found out we were making these precision animal models. And we made we were studying the disease that his group was most interested in. And in this case, it's what's this called 1466 mutation. And this particular mutation is unique in that it causes the gene to splice in the wrong places. So it's just showing you how things, are spliced together. It represents over 50 cases that have this specific mutation or in the literature have been reported. And the problem with this is that single nucleotide change mimics the normal signals that tell the gene where to splice. And so we need a way of masking this. So can we block this and then restore gene function? And that's a perfect example of how ASOs can work. The ASOs can bind to DNA in a very specific manner. And so what's you see in the lower part of these figure is if on the left-hand side, if this is what normally occurs, so normal splicing for exons 13, 14, and 15 would then put them in this order. But now this exon 13 mutation causes the gene to splice in the wrong area. And what it does is it puts the whole reading frame out of whack. And so instead of putting in the right amino acids, it then starts putting in the wrong amino acids, and then it comes to a stop. So it leads to a premature stop condition. And so what we hope to do is block that signal. So can we suppress that aberrant splicing? Now you get splicing, you still have the mutation there. And so you splice together these exons, but now instead of making the normal amino acid, it's got a new amino acid in there. But we demonstrated that that's okay. You could have that amino acid and the protein will still work. And that was a key part of our development over the years. And then we developed cell lines and made other reagents in order to test this. So we created what's called human iPS cells, and we made them with both copies of this mutation. And then we demonstrated that we could bring in the ASO, bind to this region that should not be splicing, and then we could restore the function of the gene such that it worked normally. And so this is really shown in this figure. We published this data a couple of years ago. If you take the cells and look at how much neurofibromin is normally made in these cells, you can see a large band, and this represents just in triplicate what there are 3 different independent experiments. If you take a control nucleotide, so one that does not have the exact sequence to bind properly, we can apply it to those cells and you don't see any neurofibromin made. But if we take that antisense oligo or morpholino that will mask that area specifically, now you can see we're restoring gene function and we get NF1, we get neurofibromin made again, and it changes our GTP RAS activity. And so this is just a readout of of how much neurofibromin do we need to restore in order for a gene to work properly. And so I get that question a lot, is with gene therapy, how much correction do you need to actually make the system work correctly again? And we think that that number is low. We think that if we can maybe restore as little as 10 to 20% of neurofibromin, we think that the system or the cells will respond normally. And so that's what our target really is at this point. So now what we've done is we also made a mouse. And what we did is because the mouse sequence was a little bit different than the human in this region, we had to make a mouse that had this large chunk of DNA that represents exactly the human sequence. So now when we're testing the drugs in our animals, it will mimic exactly what would go on in humans with this system. And so we've created animals that have this mutation, that have this exon 13 mutation. And this particular model is one that we're using quite a bit now. And that's because you can see that this mouse, it's called a HOXP7. And the key here is the mouse gets hyperpigmented cells. So you kind of get the cafe au lait spots in this mouse. You can also get cutaneous tumors. If we look carefully in the skin, we can see these swirls. These are small neurofibromas that are developing under the skin of this animal. And then we also get the plexiform tumors. So these are the tumors that form along the spinal cord and can impinge here. So this is the mouse model that we're using most for testing gene therapy approaches because it kind of captures multiple symptoms typical in NF1. Well, so coming back to the overall, probably your first impression of gene therapy is, how do we restore gene function? And one of the things that we try to do is, one thought is if instead of focusing on these specific mutations, can we somehow restore the entire gene back all at once? Well, the problem with that for NF1 is it's very large. So typically for these kind of gene therapy approaches, when you read about a lot of other genes, they can use a virus or an adeno-associated virus, and they can simply replace the entire gene. The problem for NF1 is it's too big to fit into those viruses that are commonly used. And there are groups now that are trying to modify the virus and stretch the boundaries a little so we can stick in NF1. And we're doing a little of that. But the other thing we're doing is using nanoparticles. So nanoparticles can carry much larger payloads. And so we're trying to stick back the entire NF1 gene, and we have demonstrated that we can create a full-length NF1 gene or what's called a mini gene. We could package it in nanoparticles. I have collaborators at Yale that are doing this, or experts in the field. And we're also trying to package the RNA and even putting back purified neurofibromin protein. We're trying different ways of packaging all of NF1 so we can deliver it in our animal models. And probably the best animal model for us to test this is in our rats that develop mammary tumors. So in this case, we have rats. And really, the whole idea for NF1 and breast cancer had been in the literature for a while, and some people were observing it, but it wasn't until my colleague, led by a graduate student, Dissinger et al., published in 2018, that if you make a rat that has a defect in the NF1 gene, just one mutant allele, the females develop mammary tumors. And they do so very quickly. And so we use this as a model system. You can see these diagrams. This is the mammary tumors and underbelly of a female rat. And normally by 16 weeks of age, 75% of our animals that have this one mutant allele will develop mammary tumors. So that's a very potent readout for us to then test gene therapy. And what we've been doing is taking our nanoparticles to deliver the full NF1 gene. And what we can see is that animals that were injected directly into the tumors with these NF1 nanoparticles, that the tumors slow their growth. That's shown in the red bar. If you just give them a vehicle or control solution, the tumors continue to grow. So this is a promising treatment for gene restoration, and we're now working with a couple of different groups, and the challenge now is finding the right nanoparticle that will target to different tissues that are affected by NF1. And So now I want to change a little bit and really talk about CRISPR and gene editing. So I think that I've shown here, the promise of gene editing, and CRISPR is working for a lot of different disorders, but there are really significant challenges for NF1. And when it comes to the reagents have to be customized for each different mutation. If you think about that, the amount of work that it goes into just trying to target one mutation is significant. I mean, it literally, we've been trying to work with 3 or 4 mutations, and it's taken us years to get something that maybe works 10-20% of the time. When you think about over 7,000 different NF1 mutations, well, until the technology further improves, then this is really, it's not going to be a good approach. And the bigger issues with NF1 and other tumor suppressor genes is you have to be nearly 100% efficient, because the problem with these technologies is they cut the DNA where the mutation is located, and then they repair it. But the problem is if you cut the DNA and you repair it wrong, you've now created a whole new mutation that you now need to target. So for tumor suppressors, it's just very different than for, let's say, a structural gene where, if you make a mistake in repairing it, it's not that big a deal. You've restored some function, and that's important. Here, for a tumor suppressor, you really have to restore all the genes, or you have to correct them all at once. So I think as much as I would like for CRISPR to work, I think it's still challenging, and it's going to take quite some time. And so trying to come back to this diagram, I've hopefully showed you that, we have a lot of different patient mutations. We have to match the kind of therapy. We try to develop cell lines and animals to develop those therapies. And then there are challenges in developing these optimal routes of delivery. And so we still are faced with a lot of challenges with NF1. And what I really want to say to this group is I'm part of a group, the RAINS Gene Therapy Working Group. They recently put out a survey. I'm not sure if any in the audience managed to participate in this. I know that we're expanding out into multiple areas, trying to reach different target groups. But it's really, it's not just what we can come up with as scientists. Can we, come up with ways of correcting the genes and therapy? But what is it the patients really want, and what is the level of risk that they're willing to take in order to undergo these trials as we start coming up with these methods? And I think it's really important to have patient engagement and for you guys to provide feedback through your constituents and really, help us understand where we're going with gene therapy and what we can do. And so with that, I'll just, to finish up and acknowledge these are a lot of the people in my group that are actually doing the work. I've pointed out my colleagues and supporters. The Gilbert Family Foundation, of course, has been wonderful to support us. A lot of this work we initiated at UAB, and Bruce Korf is the one who originally approached me and got me involved with neurofibromatosis. The Nothing Is Forever with Joseph Kammermeyer has been a wonderful collaboration. And really, I want to thank all my colleagues. Thank you for your attention today. And I'll mention one last thing is we have ongoing studies working with a group at Vanderbilt. And it's nice to see that cognitive functions are finally being addressed now, I think, with multiple groups. So I think what we're doing with neural imaging in some of our models is really going to be informative, and I'm looking forward to the next few years of therapies that may start finally addressing cognitive issues with animals, and hopefully that we relay that to patients. So thank you for your time today.